All right, this is the review section for exam 3. You can go online and find the essay questions. Again, I just want to remind you, exams aren't about who's the smartest or, uh, who is the luckiest or anything like that. It's all about being prepared, and that's the slogan of the Boy Scouts there. So spend lots of time preparing and choose the methods that work best. Those are active methods where you're actually doing something, writing, talking, trying to answer questions, working with partners. And we know repetition works, so plan to do things in short sections but to repeat them. And so those are all really important things. So the one chapter that we worked on was chapter 8, photosynthesis. And so why is photosynthesis important? It uses energy from sunlight to produce glucose. Okay, that produces almost all the food for the biosphere, all the food for humans, and over geologic time, that's produced all fossil fuels. Okay, photosynthesis also makes oxygen gas. That's all the oxygen in the atmosphere. There's no other process that does that, and that's important. And it also makes cellular respiration possible. And now we're talking about carbon emissions and how fossil fuel burning is damaging the atmosphere. That the um, removing carbon dioxide from the atmosphere, there is a way to do it, and that's through photosynthesis. Okay, so photosynthesis involves two main sets of reactions, the light reactions, and it should be the Calvin cycle, not the calving cycle. The Calvin cycle. Oops, that's a mistake. And these things occur in chloroplasts and in some bacterial cells. In eukaryotes, this is in plants, and it's all in chloroplasts. And so the light reactions occur in the membranes of the chloroplasts. So that's chlorophyll and ATP synthase. These are embedded in the membranes. The inner membranes are thylakoid membranes of the chloroplast. The light reactions capture photons from sunlight. They use it to split water to make oxygen. And the energy that's gotten from these electrons is used to pump hydrogen ions through the membrane, and those are allowed to diffuse back through by ATP synthase, and so that makes ATP. Okay, photosynthesis uses the pigment chlorophyll to absorb energy from photons. Okay, that energy it absorbs comes in, the chlorophyll absorbs energy from red light and from blue light. It appears green because it reflects green light. Okay, the Calvin cycle, that's the second part. These are the dark reactions. They occur in the fluid part around the thylakoid membranes. And here, these Calvin cycle reactions use ATP and NADPH from the light reactions, and they take the carbon from carbon dioxide and they attach it to a molecule called PGA, or they make PGA out of it. And this uses an enzyme, Rubisco, and Rubisco is really important. And doing this a bunch of times allows the Calvin cycle to make glucose. And so this is where it happens. Um, there's some alternate ways that plants fix carbon. Um, and these are because the regular form of photosynthesis, which is most plants, isn't so good in hot and dry climates. The plants have to open pores to let in carbon dioxide from the atmosphere, and in hot, dry climates, there's a lot of water loss through the pores and through the regular form of the Calvin cycle. So some plants modify the Calvin cycle. In corn and sugarcane, these are called C4 plants. They use an enzyme to make a 4-carbon molecule from CO2, not the 3-carbon molecule. And they do this in a separate cell, so they don't have to have their stomata open as much. And so this uses slightly more energy, but if it's very hot and dry, this allows them to do photosynthesis when the other plants will be wilting. In even drier environments, they use another form of metabolism called CAM. These are found in this type of metabolism is found in cacti and pineapples, and they open those pores, the stoma, only at night. And so they fix CO2 at night using stored energy, and they make it again into a 4-carbon molecule. Again, this uses more energy, but there is almost no water loss. Okay, so then we went into cell signaling in chapter 9, and there are 4 basic mechanisms cells use to communicate with other cells. These include direct contact, where the cells are in physical contact. They include paracrine signaling, where the cells are a short distance away and they release a chemical into the fluids around them that will affect the nearby cells. In endocrine signaling, animals and some plants release chemicals that act at distant cells, and this chemical goes through a long transport, maybe in blood or other fluid, and it can act at distant cells by acting with receptors. And in some animals, or in animals only, there's synaptic signaling, where specialized cells have long extensions that release a special chemical, a neurotransmitter, at distant cells. In all cases with this cell signaling, there's a ligand that's being released. These ligands have to bind to receptors. Some receptors are on the surface of the cell, some receptors are inside the cell. Okay. In intracellular receptors, the ligands move through cell membranes and they bind to molecules inside the cell. And these are protein receptors that are then going to go to the nucleus and turn genes on or off. Other receptors are on the surface of the cell. These other receptors are 3 types. There's ligand-gated ion channels. So the ligand binds to the ion channel, kind of allowing it to open, and so ions can move in and out of the cell. Then there's enzymatic receptors, which are mostly proteins that can transfer a phosphate to and from other proteins. And then a specialized kind of enzymatic receptor is the G-coupled receptor, which transfers a molecule from guanine triphosphate, or GTP. And so this is a special kind of phosphate receptor, very, very common. Okay, cell-to-cell communication is really important. This is what lots of medicine is based on. We treat most things by altering cell signals. And so we may give a molecule that looks like a hormone or looks like a neurotransmitter to help change what cells are doing. And this is particularly true for something like insulin, where we're treating diabetes these ways. This is a huge thing. All these people used to die. We treat lots of things by doing this. This is also drugs of abuse are often drugs that affect cell signaling. And so like smoking and nicotine. Nicotine resembles a neurotransmitter called acetylcholine. And people smoke so that they can get this nicotine, and that simulates the effect of acetylcholine in the brain. And so this is typical. And this has some drawbacks because what happens is people start making fewer of these receptors, and so they need an extra way to get it going, and so that means they become dependent on the drug and they can tolerate higher and higher doses. And if you take the drug away, they will have withdrawal symptoms. So it's serious. And so lots of drugs that are abused are drugs that are simulating chemical signaling between cells. So cocaine, the opioids, the amphetamines, they all do this. They're all working on these cell signaling pathways. Okay, then the last 2 chapters involved cell reproduction. In chapter 10, we talked about how prokaryotes reproduce, and they use a method called binary fission. In binary fission, prokaryotes have a single circular chromosome. Replication starts in one spot and it proceeds around the chromosome. And then as it's completed, the chromosomes, each separate chromosome becomes anchored to the cell wall or cell membrane in a different place, and a septum forms between the 2 chromosomes by this protein FITZ. Okay, this just isn't possible for eukaryotes. Eukaryotes use mitosis and cytokinesis. They have multiple chromosomes, and the chromosomes the DNA molecules are wrapped around histones, and then they use some microtubule proteins, cytoskeletal proteins like microtubules to do this. And eukaryotes have to do this because they have huge chromosomes and a huge amount of DNA. These chromosomes are much larger than anything that prokaryotes have. Eukaryotes have multiple chromosomes, always in pairs, 2, 4, 6, 8, something like that, and literally billions of nucleotides, 1,000 times or more the DNA of a prokaryote. And so, this DNA is almost always associated with proteins, and so they just can't do cell reproduction in binary fission. It wouldn't work. So they use mitosis. They have these duplicated chromosomes attached together at a centromere. The centromere becomes attached to kinetochore microtubules, which become attached to a spindle. They line up all the chromosomes at the center of the cell, and half of each one goes in the opposite directions. And so it's a simple way to work with all this DNA and all these chromosomes. Okay, this is highly controlled process. We've looked at the life cycle of the cells. This is the cell cycle here, and cells have certain chemicals that are associated with the cycle, and certain chemicals that are associated with each step in how cells go to reproduce. Many of these chemicals are called cyclins, and many of them transfer phosphates, so they're cyclin-dependent kinases. And at certain checkpoints, these proteins have to become active. Okay, and so these cyclin-dependent kinases act at these checkpoints to control cell division, to make sure DNA is okay, that the cell is ready for reproduction, and they can stop it if the cell is not ready. And so we've talked about some of these in detail. These have a lot of important applications. When we study the cell cycle, we can sometimes, when something goes wrong, that leads to cancer. And so one of the main applications is understanding the changes that happen in the regulation of the cell cycle that lead to cancer. But this is also important for healing and regeneration. And so things like spinal cord injuries, traumatic injuries to the heart, transplant patients, degenerative diseases, all of these things can benefit from our learning control of the cell cycle. And one of the things we've figured out is that some of these proteins that act at these checkpoints are really involved in preventing cancer. And cancer often occurs when these proteins are damaged and don't do their normal job. And these are called tumor suppressor genes. And they act like the brakes on the car, in that they're like, if they're damaged, the cell reproduction will go faster and faster. And if one tumor suppressor gene is damaged, it's not too bad because you still have another copy. But if they're both damaged, then the cell cycle can go on in an uncontrolled manner. And so that leads to cancer. We talked about two of these genes, P53 and the RB gene, but there's many of them. Okay, then the other type of cancer-causing genes are the proto-oncogenes. And there's a larger number of these, and they're all proteins that are involved in growth factor signaling. And so they're involved with promoting growth. And if one of those is damaged, it's like a growth signal that's always on. Okay, so then we also talked about another type of cell reproduction that's involved in eukaryotes, and that's meiosis. And meiosis is part of the life cycle of sexually reproducing organisms. And so these organisms have part of their life cycle as diploid, where they have two copies of each chromosome, two copies of every gene. And then the other part of their life cycle is haploid, where they have cells that have only one set. Okay, and so these are like the sperm and the egg. And so when the sperm and the egg fuse, the offspring inherit genetic material from two parents, and will have two copies of every gene. This is important, so it allows you to have new offspring that have inheritance from both parents. And there's some slight differences between meiosis and mitosis. There's a lot of similarities, but one of these features is that in part of meiosis, an early part in prophase 1, that homologous chromosomes line up next to each other. They both carry genes for the same traits, and sister chromatids join and remain joined at their centromeres during this process, and they can exchange segments. Okay, and this is a really important thing. And then instead of in mitosis, where individual chromosomes migrate by themselves to the metaphase plate in metaphase and are split. In meiosis, there is no splitting of the centromeres in meiosis 1. That only happens in meiosis 2. And so here we have meiosis 1 and mitosis compared. And so you can see homologous chromosomes line up. The homologs align themselves on the metaphase plate. And in telophase 1, whole separate chromosomes have moved. And so the chromosome number is reduced. Okay. And then in meiosis 2, we have just individual chromosomes lining up on the metaphase plate and then the centromere splitting. Okay, this is really important for eukaryotic organisms because, why have sex? Well, the answer is if you have sex, you can have 2 genes for each trait, and they can likely be different genes. And so if one is bad, you still have another copy, and that copy may work well, or it may work well under certain conditions. And so you have this ability. And because you have inheritance from two parents, and that inheritance involves scrambling their genes, the offspring have lots and lots of genetic variability. Okay, so check over the essay questions on the handout and look over at the practice exam. I will post a corrected version of that on Saturday, and so you can look at that, work on it on Friday, and you'll see it on Saturday.